Biodiversity. Species is a distinct population. Within the species, subspecies, types, and variations exist. As a result, a wide varieties of organisms exist on the Earth's surface. Existence of varieties of life forms on Earth's surface is called biodiversity. The term biodiversity is often replaced by the term species diversity. The species diversity is number of different species and abundance of each species present in a given community. There is another term called species richness. Species richness is number of species in a given community. Biodiversity is not evenly distributed on Earth's surface. Biodiversity increases from the polar region to the tropic. Tropic is the regions near the equator. Increasing biodiversity from polar region to tropic is called latitudinal diversity gradient. Diversity increases with the increase in temperature. Marine biodiversity is high in Western Pacific region where temperature is high. Biodiversity hotspots. The hotspot concept was introduced by scientist Norman Myers. According to him, biodiversity hotspot is a biogeographical region with biodiversity under threat. A region must qualify two criteria to be recognized as biodiversity hotspot. These two criteria are: the region must have at least 1500 species of endemic vascular plant. Endemic species means the species restricted to a geographical area, and the region must have lost 70% of its primary vegetation. This means the region must have some threatened species. Total 36 regions of the Earth's surface qualify these two criteria. So, worldwide, there are 36 biodiversity hotspots according to the criteria set by Norman Myers. Biodiversity hotspots of India. 4 out of 36 biodiversity hotspots are represented by different geographical regions of India. The Himalaya hotspot region is divided into two parts, Western Himalaya and Eastern Himalaya. A part of Indo-Burma hotspot region is represented by Northeast India, Western Ghat hotspot region, and Sundaland hotspot region, which is again represented by Nicobar Island. Each of the 4 hotspot region has different climatic condition and different biodiversity. They also have their own issues to be addressed. Every hotspot zone has a list of endemic and threatened species. Measure of biodiversity. Biodiversity means variation of flora and fauna. Variation of flora and fauna is measured in 3 levels. Ecosystem level. At this level of study, diversity of different ecosystems is studied. For example, aquatic ecosystem, desert ecosystem, grasslands ecosystem, forest ecosystems. These are the different ecosystems may be present in study area. Next level of study involves the study of diversity in species present in each of these ecosystems. So for example, in grassland ecosystems, the species 1, species 2, species 3 and species 4 are identified. And third level of study is the genetic level. At the genetic level, the diversity of gene or the variations in gene is studied. So these 3 levels of variation can be understood by an example. So let us understand it organically. Suppose there is a study area and this study area has got different ecological regions like forest, desert, grassland and lake. Each ecological region represents an ecosystem. So these are documented as forest ecosystem, desert ecosystem, grassland ecosystem and aquatic ecosystem. Existence of different ecosystem in study area is called ecosystem diversity. In the next level of study, species present in different ecosystems are documented. Existence of different species in different ecosystem is called species diversity. The third level of study is documentation of variation in genes. The variation in the same gene is called genetic diversity. To understand this genetic diversity, let us take an example. For example, there is a population of plant species growing in this forest area. Some individual of these species bear white flower and some individuals bear red flower. The presence of two different flower color is due to presence of two different alleles for the same gene. This means the gene responsible for the color of the flower is having two alleles. One allele is for the white flower and the other is for the red flower, creating two varieties of the same species. So variation in gene is called genetic diversity. So these three levels of variation are documented to describe the biodiversity of a given region. Once we document the biodiversity at three different levels, that is ecosystem diversity, species diversity and genetic diversity, we need to calculate the diversity index. Diversity index is the quantitative measure of biodiversity, or here we can use the term species diversity in place of biodiversity. And the index is a numerical figure that explains the biodiversity of a study area. The diversity index is calculated using different methods. There are more than one way to calculate biodiversity index. However, we have mentioned only two very important diversity index. They are the Shannon-Weiner index and the Simpson diversity index. Both the indices are calculated by their respective formulas. The calculation of Shannon-Weiner index is based on two factors. One is species richness and other is species evenness. Species richness is the number of species present in a given community, and the species evenness is relative number of individuals of each species in a given area. The species richness and the species evenness are calculated from the list of species and then easily put into the formula to obtain the biodiversity index. Shannon-Weiner index is influenced by the species richness, that is abundance of a species, and evenness of the distribution, that is the presence of dominant and rare species. So it is sensitive to even a small change in both the factors, and it is popular and widely used to determine the state of the ecosystem. On the other hand, the Simpson Biodiversity Index is based on the probability that two individuals collected randomly belong to two different species. The Simpson index is influenced by dominant species, so it is used to determine the dominance of species that shows the trend of the ecosystem diversity. Both the indices are used to obtain the information that are useful to measure biodiversity of a region. So there is one more way to measure the biodiversity. The ecologist often observed that the biodiversity is not evenly distributed. The biodiversity often exists in patches or confined to certain localities. So alpha, beta and gamma diversity were introduced by R.H. Whitaker to measure biodiversity in a given area where the whole area is divided into some localities or sites or ecosystems. The total species diversity in a given area is called the gamma diversity. The gamma diversity is determined by two other findings. These two findings are the diversity in different locality or site or ecosystems, and that is called the alpha diversity. And the second one is the differentiating among these sites, that is the comparison between the two sites. It is called the beta diversity. The diversity within a locality is called alpha diversity. The alpha diversity is expressed by number of species in each locality or site or ecosystem. That means it tells about the species richness in that site or locality. For example, in a given area, there are 4 different locality or sites. Each locality may be represented as an ecosystem, and each ecosystem has species diversity. Species diversity of each site or locality or ecosystem is the alpha diversity of that site or ecosystem. So the alpha diversity of site 1 is 2, alpha diversity of site 2 is 3, alpha diversity of site 3 is 2, again alpha diversity of site 4 is 2. Let us take help from a table. Here in this table, the species A is present in site 1, 2 and 3. Species B is present in site 1 and site 2. Species C is present in site 2, site 3 and site 4. Species D is present in site 4 only. So alpha diversity of each site can be easily calculated from this table. So next point is the differentiation among these sites. The differentiation of these sites, the beta diversity is change in species diversity between two ecosystems. The beta diversity is obtained by eliminating the common species from both the sites. So if we compare the site 1 and site 2, the beta diversity is 1. So if we compare the site 2 and site 3, the beta diversity is again 1. And if we compare the site 3 and site 4, the beta diversity is again 2. So we can compare any two localities to obtain the beta diversity. And finally, the total number of species represent the gamma diversity. So the gamma diversity is 4.